I think so. We'll move to the next speaker, Dr. Suresh Damodharan, who is going to speak on the interchangeable biosimilar insulin, evidence and benefit. It's a pleasure to introduce Dr. Suresh Damodharan, who is a consultant, diabetology, thyroidology, thyroid, and endocrinology at Sri Ramakrishna Hospital, Coimbatore. Over to you, sir.
Good evening, everyone. Can you all hear me?
Yes, sir.
Fantastic. Thank you very much, Praveen, Dr. Praveen, and also all my colleagues. The next 15 minutes or so, if I can touch base on the clinical trials, basically, if you take US FDA or European body, all needs a very strict, stringent clinical trials. That I think that's what Manoj was mentioning very early. And also, if you look at, for example, the EMA, which is a European body, they need a PK/PD study and a phase 3 trial, which is needed for type 1s. And if you look at the FDA, which has got pharmacokinetic and pharmacodynamic study, along with that, I think you need a type 1 and type 2 study with interchangeability. So it is a very strict bodies. They need clinical trials. So if I can touch base on that, we know for glargine as a molecule, it has got a huge trial, but when you come to biosimilar, it is, as I mentioned, because of the very stringent things for all the bodies, it has to be the whole global trial has to happen. And what happened with the Biocon molecule, the biosimilar, there is all the clinical trial happened throughout the world, and India approval happened in 2009. Whole of the world, there are so many countries have taken up the molecule. When we are training first, and then Europe, these molecules were first came in. That is a very long time ago. But if you look at the clinical trials as a whole, the PK/PD trial of a phase 1 of a Baselog versus Lantus, which is available in Europe, and also US availability in Stride 1, 2, 3 actually. So 2 is with the type 2 and 3 with type 1, again with the switch study. I'll just come to that and touch base on that. So throughout the world, this global trial happened. And you look at the Slovakia, South Africa, if you go to the European countries, and also if you come back to Africa, and also Australia. So it has happened throughout the world, especially Japan has got a very, very strict body. First, I think Biocon's biosimilar has got approval in Japan, if I can remember properly. So almost 7 clinical trials, and with almost whopping 2,000, almost 2020 countries. So if you take the first one, which is needed for the European body, which is the type 1 diabetes, which is the proposed biosimilar of glargine of Biocon's one with the reference insulin in a type 1 diabetic patient. So we are checking about the PK/PD, and the study population is around 114 patients. Now listen, this is very important. A single center trial. It is a double blind, and also it is hyperinsulinemic euglycemic clamp with patient receiving almost 0.5 units per kilogram of each drug. If it is 60 kilograms, then it's almost 24 units. So YMYL is the Biocon biosimilar drug, and then the US glargine and the European glargine. There is 28 periods, 28-day period. It is a single trial, single center trial, with double blinded, and it's a three-way crossover with the clamp. So the first clamp, the first visit, and also on up to the 5th visit for a follow-up. Now you look at the pharmacokinetic comparison. So the end of the trial, the blue one is the Biocon's biosimilar one. The light blue one, you can see it is almost 100% comparable with the original molecule of glargine, which makes it much more sort of plateau insulin, like the original insulin itself. So the GIR profile is also very, very smooth. And if you look at the conclusion of this trial, it is both PK/PD endpoints is almost having a bioequivalence, and biosimilar drug has got very, very well tolerated, and no significant safety issues has emerged for the type 1 diabetic patient in a single trial control in a hyperinsulinemic euglycemic clamp. So that is number 1 trial. So if you go to the InStride 1, which is the second trial with the non-inferiority study, to compare the efficacy of the safety of the Biocon insulin with the glargine insulin in a type 1. So again, this is a type 1 patients. If you look at it, it is compared with head-to-head with, and it is published in 2017 ADA. The study design is a multicentric trial, and it's a phase 3 with type 1 diabetics around 558 with A1C below 9.5. And the age group is between 18 to 65, and screening is 6 weeks, 4 weeks, run-in is 6 weeks, and then the follow-up after the 52 weeks is for 4 weeks. So this is the head-to-head with the Biocon glargine with the other molecules in combination with bolus insulin as well. So you look at the primary endpoint, the change in A1C is the main thing, but the secondary endpoint is the A1C change and also the fasting and the SMBG. And the secondary is the hypoglycemia, which is very, very important. The nocturnal hypo and the hypo, and then the immunogenicity, because people, that's what they say as the immunogenicity. I'll come back to that in the last clinical trial. So if you look at the actual A1C and actual fasting and the visit of treatment, almost A1C stabilized from the baseline. The actual fasting glucose on the visit treatment was almost the same. So that concluded the second trial with the InStride 1, showing that Biocon's insulin and the reference insulin was almost similar at the wake of 24 to 52, almost a year, and there is no statistically significant A1C reduction. So that tells you the drug stays in a pharmacokinetic and pharmacodynamic profile, and also it is efficacious. So you can claim that if it is there, and also it is not efficacious, but it is efficacious. That is both basal and bolus insulin level did not differ from between the groups, and the rate of hypoglycemia is also comparable. That's what, honestly, you have to say that honestly we can see that in the clinical practice as well. The InStride 2 is another trial, which is the type 2 diabetics, compare with the efficacy of the safety Biocon. Just to show, this is a multi-centric trial in type 2 diabetics, almost 560 patients. You need a better screening and stable OAD, so plateau with the OADs before getting into the insulin. So you can see the biosimilar glargine and the reference glargine with 24 weeks with the numbers of 277, and then the follow-up for almost 4 weeks. So 4 weeks of screening, 24 weeks, and then followed up by 4 weeks of follow-up. So this is to show the primary endpoints is A1C, just to see the effectiveness. And also, if you look at the secondary endpoints, you look at the change in fasting blood sugar, insulin dosage. If the competitor is using less insulin compared to the biosimilar insulin, and as I said earlier, the very, very important thing is the safety, which is the hypoglycemia and the immunogenicity. And what happened in the clinical trial, you can see the blue line and the reference glargine, both are exactly the same. This is almost like a 24 weeks. You can see the blue line, both are almost completely controllable. That is in the A1C, and you can see that there is no statistical difference at all. So if you look at the dosage as well, it is not like that you have to use a high dosage when it comes to the biosimilar insulin. It's not like that. You can in the clinical trial of InStride 2, it is almost the same dosage. So just to conclude of the InStride 2 trial, this mean A1C reduction was almost the same, and it is non-inferior to the insulin glargine, and the fasting blood sugar changes and the hypoglycemia rate is almost comparable with the both groups. So I have spoken upon InStride 1 and 2, and also the type 1 diabetic patients with the PKPD trial. So all this has been spoken. And then the last clinical trial is the InStride 3. It is to show not only the efficacy, and we spoke about safety, and then the immunogenicity, which is very, very, very important. When people of type 1 diabetes switched between Biocon's glargine and reference glargine. So what happened in this? You can see the type 1 who are completed the INSTRIDE 1 or on the reference stage almost 52 weeks, you randomize them into Biocon's insulin glargine with Lispro as a basal bolus regime, and then the reference insulin with basal bolus and the Biocon insulin switch over again. And what happened after the 36 weeks clinical trial and then the follow-up of 40 weeks. So it is the switchover trial, robust design. It has been very well designed trial, and it's been published in 2020. So what happened in this? The efficacy wise, number one, the A1C reduction. If you switch it, the change in A1C baseline is to week 36 of Biocon glargine was equivalent to the reference insulin. So just to show the safety wise, hypoglycemia almost comparable with the both sequence of throughout the study, and the immunogenicity data, which I was mentioning earlier, I'll show later. It is showing that safety and immunogenicity of Biocon similar glargine is almost the same. So that brings just to show the assessment of immunogenicity, because that is very important. People claim, and that is the difference between the originality molecule and this one. But I think the incidence and change from baseline is relatively levels of anti-drug antibodies when compared between glargine and reference group insulin. So this is like a change in cross-reactive ADA, no statistical difference. It has been there with INSTRIDE 1 and 2 at any given point. So that is very reassuring. And total cross-reactive insulin antibody response, it is all inferred to immunogenicity, but I think it is similar in both the groups of the original insulin compared with the blue of the biosimilar group. So what happened during the conclusion with the immunogenicity assessment is both are comparable. Majority of patients are negative with almost like with immunogenicity, and proportions of tested positive for anti-HCG anti-HCP antibodies were similar between these two groups, indicating that both the original molecule and the biosimilars have got equal sort of immunogenicity. In summary, I think just to show, I have summarized with the first clinical trial with PKPD saying it's a head-to-head comparable, and also I've showed you with the type 1 trial of INSTRIDE 1 showing the efficacy and safety with the hypoglycemia is almost same with the biosimilar of Biocon molecule compared to the originator, and then the INSTRIDE 2 with the type 2 diabetics with a decent number of around 500. It shows in a multicentric trial that it shows that it is not only the safety and efficacy, but also the fasting blood sugar also was very good when almost head to head with the original molecule. And then the INSTRIDE 3 and also the immunogenicity data suggests that even for the type 1 with INSTRIDE 1 and 2, if you check the immunogenicity, both are comparable with the original molecule and the biosimilar. So that takes us, this is the world first interchangeable biosimilar insulin. Manoj was mentioning about interchangeability in a Western world. I worked when the growth hormone days of biosimilar came in. He's absolutely right. I think when the clinical pharmacist comes in and checks whether the original molecule is there or the Omni, the biosimilar is there, and they can relatively, when you got a data of less immunogenicity and same efficacy, they change it because of the cost and made it in India, and it's a high quality insulin. So I stopped there just for an HCP to provide a quality is a very important thing. I think Manoj has touched base and effectively mentioned all throughout his talk. So I stopped there and happy to take any question.
That's great. Thank you very much. And you summed it up very nicely.